Off the Galápagos Islands in 1987, researchers Tom Arnbom, Hal Whitehead, and Jonathan Gordon recorded something specific. A pod of sperm whales going completely silent as killer whales closed in on them. Not a defensive click burst, not an alarm call. Silence. The whales held position, tails outward, and waited. Only when the orcas began circling directly did they produce what Arnbom and colleagues described as intense bursts of clicks. The author's interpretation was measured. Those clicks were most likely echolocation, the whales trying to locate the threat, not an offensive acoustic discharge. That paper is 38 years old. The claim built on top of it that sperm whale clicks function as acoustic weapons capable of injuring or killing is considerably more recent and considerably less careful. The clicks of Physeter macrocephalus are the loudest biological sounds ever measured. That part is true, and it's enough to make any number impressive. But extraordinary output and demonstrated biological effect are two different things. 4 claims about these clicks will be tested here, in ascending order of difficulty, each ending with a verdict the evidence can actually support. Before any number can be evaluated, the mechanism needs to be understood. The sound starts deep inside the sperm whale's head at a structure called the phonic lips, also known by the French anatomical term museau du singe, monkey's lips, located at the anterior end of the right nasal passage. A single pair, not the 2 functionally separate pairs found in most toothed whales. Air from the pressurized right nasal passage is forced through those lips, causing them to vibrate and generating the initial acoustic pulse. Ted Cranford's 1999 paper in Marine Mammal Science, still the canonical anatomical reference for the sperm whale's nose, described the nasal complex as a bioacoustic machine operating through this pneumatic mechanism at the monkey lips. What happens next is the part that makes sperm whales unusual. That initial pulse doesn't travel directly into the water. It propagates backward through the spermaceti organ, a wax-filled structure that can stretch for 2/3 of the animal's total body length in large males, until it hits a reflective frontal air sac against the skull. The pulse reflects forward through a complex of fatty, lens-shaped tissue called the junk, which focuses and collimates the sound into a narrow forward beam. Walter Zimmer, Peter Tyack, Mark Johnson, and Peter Madsen confirmed this geometry in a 2005 study in the Journal of the Acoustical Society of America. Their 3-dimensional beam reconstruction showed a narrow, forward-directed beam with a directivity index of 26.7 decibels, plus a broader, backward-directed component and a low-frequency omnidirectional signal from air resonance. They called this the bent horn model. Sound channeled rearward through the spermaceti organ, reflected at the frontal sac, projected forward through the junk, like a bassoon in reverse. The anatomical consensus on what this structure evolved to do is consistent. Every major peer-reviewed study of sperm whale bioacoustics describes the spermaceti organ, phonic lips, and junk as an integrated biosonar system, optimized for echolocation and communication, not for offensive acoustic output. That's the biology. Now the number. In 2003, Bertel Merle and colleagues at the University of Aarhus published what remains the definitive source-level measurement of sperm whale clicks in the Journal of the Acoustical Society of America. Using a large aperture hydrophone array, they back-calculated source levels for a limited number of assumed on-axis clicks and reported a maximum of 236 dB re 1 micropascal RMS at 1 meter. Their statement in that paper was unambiguous. The sound levels measured make sperm whale clicks by far the loudest of sounds recorded from any biological source. A 2000 paper by the same group had reported peak source levels up to 223 decibels, right 1 micropascal, with level differences of 35 decibels between on-axis and off-axis recordings of the same click. confirming the extreme directionality of the forward beam. The figure 230 decibels that circulates widely is a rounded approximation. The measured maximum from Merle et al. 2003 is 236 decibels. The rounding costs some precision, but the more consequential problem is what precedes the unit. Decibel right 1 micropascal. Decibels reference to 1 micropascal is the standard unit for underwater sound pressure. Airborne sound uses decibel reference to 20 micropascals, which approximates the threshold of human hearing at 1 kilohertz. Different reference pressures, different propagation media. Converting between them is mandatory for the comparison to mean anything. The reference pressure difference alone accounts for about 26 decibels. Water and air also differ substantially in acoustic impedance. Water's characteristic impedance is roughly 3,600 times that of air at sea level. Accounting for both the reference pressure and the impedance ratio, a conversion of approximately 61.5 decibels is required when comparing equal acoustic intensities across the two media. Southampton's Institute of Sound and Vibration Research has documented this. To convert an underwater measurement from decibel right 1 micropascal to an air equivalent level, subtract approximately 61.5 decibels. Applied to the Merle figure, 236 decibels right 1 micropascal underwater corresponds to roughly 175 decibels in air equivalent terms. Still extraordinarily loud, but no longer a figure that occupies the same numerical space as airborne references. The viral version of this claim treats them as equivalent. They aren't. One more caveat. The 2003 paper itself provides. The 236 decibel figure isn't a direct pressure measurement taken 1 meter from the whale's head. It's a back-calculated source level. Researchers measured clicks at distant hydrophones, solved for the whale's position using time of arrival differences, then mathematically propagated levels back to a nominal 1 meter reference using assumed spherical spreading. Merle et al. 2000 acknowledged explicitly that the main source of uncertainty lies in the geometry of the recording situation rather than in the equipment. Formal error bars on the maximum weren't provided. This doesn't invalidate the measurement, it contextualizes it. An extraordinary back-calculated source level measured in underwater pressure units for a narrow forward beam produced by a highly directional biological sonar system. That's what the number describes. Now the 4 tests. Test 1. Communication. Sperm whales produce 4 recognized click types through the same phonic lips and spermaceti organ. Usual clicks serve as long-range biosonar pulses during foraging dives, with interclick intervals of roughly 0.5 to 1 second. Creaks, sometimes called buzzes, are rapid sequences used during prey capture, with intervals dropping to about 20 milliseconds. Slow clicks are produced mainly by large males over interclick intervals of 5 to 7 seconds. And codas, short, stereotyped sequences of 3 to 40 clicks lasting under 3 seconds, produced primarily near the surface during social periods, rarely during deep foraging dives. In 1993, Linda Weilgart and Hal Whitehead published a study linking coda production to specific temporal and social patterns in stable female groups, concluding that codas function primarily to maintain social cohesion after foraging bouts. A decade later, Luke Rendell and Whitehead published data from 18 social units and 61 groups recorded across the South Pacific and Caribbean between 1985 and 2000. The coda repertoires clustered reliably into 6 acoustic clans. Clan ranges spanned thousands of kilometers. Different clans occupied the same ocean regions simultaneously. Rendell and Whitehead's conclusion: culture may thus be a more important determinant of sperm whale population structure than genes or geography. Genetic analysis supported this. Sympatric clans share many mitochondrial DNA haplotypes, meaning kinship does not predict coda similarity. The dialects are learned, not inherited. A 2022 study in PNAS confirmed that certain coder types, identity coders, are used frequently within clans and rarely by members of other clans, functioning as acoustic markers of group membership. A 2025 preprint by Gaspar Begusch and colleagues found vowel-like and diphthong-like spectral patterns in sperm whale coders, concluding that sperm whale coders represent one of the closest parallels to human phonology of any known animal communication system. That paper is a preprint and not yet peer-reviewed, but the communication function it builds on has decades of independent corroboration behind it. Verdict: established. Sperm whale clicks, specifically the coder type, function in social communication. Multiple independent peer-reviewed studies confirm this across populations, clans, and decades of field observation. Test 2. Echolocation. A foraging sperm whale begins producing usual clicks at depth around 100 to 220 meters during descent and continues to foraging depths typically between 400 and 1,200 meters. Total darkness under crushing pressure. Echolocation isn't merely useful at those depths, it's the only viable sensory mode for detecting prey. Patrick Miller, Mark Johnson, and Peter Tyack published a tag study of 23 sperm whales in 2004 in the Proceedings of the Royal Society B. Their data showed that 99.8% of creak sequences, the high-repetition terminal sequences used at close range, occurred in the deepest 50% of each dive, and 57% in the deepest 15%. Creak rates correlated directly with time spent at maximum foraging depth, and the pattern matched bat echolocation during prey capture, fast repetition at close range, slower intervals during search. In 2020, Pernilla Tonnesson and colleagues published the first direct recording of prey echoes from a tag placed on a sperm whale's nose tip. The data confirmed that whales detect single targets at ranges up to 144 meters ahead and schools of organisms at up to 245 meters. Less than 10% of detected organisms were targeted, and buzzes began more than 20 meters from prey. The study's explicit conclusion: powerful clicks enable long-range echolocation and prey selection. The acoustic system is doing exactly what a biosonar system does. Verdict: established. Sperm whale clicks function in echolocation. The evidence includes direct prey echo detection, depth-correlated behavioral sequences matching bat sonar models, and peer-reviewed confirmation from nose-tip tag field studies. Test 3. Orca deterrence. This is where the science requires the most precise language, because the question that generates the most interest, do sperm whales use clicks to deter killer whales, contains two different claims that need to be kept separate. Orcas do predate sperm whales. In October 1997, approximately 35 killer whales attacked a pod of 9 sperm whales, roughly 130 kilometers off the central California coast over 4 hours. Robert Pittman and colleagues documented the event in 2001. One adult sperm whale was killed and eaten, others were seriously wounded. The orcas used a wound and withdraw strategy in waves of 4 to 5 animals. Pittman's paper described the sperm whale's main defensive behavior as a formation, heads together, tails outward, and noted, the sperm whales appeared largely helpless. This is the baseline. Orcas can and do successfully predate sperm whales. They aren't categorically deterred. Returning to the 1987 Galápagos observation, Arnbom, Whitehead, and Gordon recorded sperm whales going silent before and during flight from attacking orcas, consistent with acoustic concealment, then producing click bursts once orcas circled or attacked directly. The authors attributed those bursts to echolocation, the whales locating the predators. The clicks served a sensory function in the interpretation of the researchers who observed them, not an offensive one. What the literature documents consistently is that adult male sperm whales represent a particularly risky and energetically costly prey target. Field reviews note that adult males are rarely, and in the documented record perhaps never, successfully killed by orcas. The peer-reviewed explanation focuses on physical risk and caloric cost. Large adult males are dangerous to attack, and the energetic payoff is unfavorable compared to smaller cetaceans or calves. That's an energetics and injury risk argument, not an acoustic deterrence argument. No peer-reviewed field study has isolated sperm whale click intensity as the specific variable causing orca avoidance. No study has demonstrated reduced orca attack success as a function of click exposure. In observed interactions, the defensive formation, tail strikes, size, and clicking occur simultaneously. Separating the acoustic contribution from the physical contribution isn't possible from field observation alone. The word terror does not appear in primary literature as a descriptor of orca responses. In 2025, Cosentino and colleagues published observations of 4 interactions between orcas and adult male sperm whales in Norwegian waters, recording codas and click bouts with varied repetition rates during encounters. That's new field data. It doesn't resolve whether clicking caused orca disengagement because there is no control condition. Verdict: plausible but unverified. Orca avoidance of adult sperm whales is documented and consistent. Physical risk and energetic cost are the peer-reviewed explanation for that avoidance. Acoustic deterrence specifically, the hypothesis that click exposure is the causally active variable, is consistent with some field observations, but hasn't been tested in isolation by any published study. Test 4. Injury and lethality. No peer-reviewed study documents sperm whale clicks causing ruptured eardrums, internal organ damage, or death in any organism, humans, other marine mammals, fish, or invertebrates, at naturally occurring exposure distances. That absence is the finding. Multiple research groups have examined this question directly. In 2006, Peter Madsen, Peter Tyack, and colleagues published Testing the Odontocetes Acoustic Prey Debilitation Hypothesis, No Stunning Results, in the Journal of the Acoustical Society of America. They exposed fish and squid to high-intensity sound pulses at intensities comparable to sperm whale clicks. The prey continued swimming normally. The conclusion was explicit. The hypothesis that acoustic signals of odontocetes alone can disorient or stun prey can't be supported. The deeper counter-evidence comes from what sperm whales actually do during prey capture. During the terminal buzz phase, the moment of closest approach before a strike, whales reduce their click source levels by 20 to 40 decibels relative to their maximum output. They aren't deploying maximum acoustic power at the kill. They're reducing it, switching to a high-repetition, lower-amplitude signal optimized for high-resolution short-range tracking. The 2016 study by Andrea Fays and colleagues confirmed this with tag data. Estimated received levels at prey during buzzing fall below known debilitation thresholds. The physics of acoustic tissue injury is real and documented, but for different sources and different conditions. Intense underwater sound can rupture gas-filled organs, damage hearing, and cause hemorrhage at sufficiently high pressures and energy densities. These effects appear in blast wave studies and in research on military sonar exposure at close range. But those injury scenarios involve waveforms, energy densities, and distances that don't correspond to what naturally occurring sperm whale clicks deliver at realistic interaction ranges. The arithmetic is straightforward. The mole source level of 236 dB, or 1 micropascal, applies at 1 meter from the whale, a back-calculated figure as established. Underwater sound spreading follows a spherical loss rule, roughly 20 decibels of transmission loss for every 10-fold increase in range. At 10 meters, approximately 20 decibels lost from the source level. At 100 meters, approximately 40 decibels lost. A received level around 196 dB are 1 micropascal at 10 meters, dropping toward 156 dB are 1 micropascal at 100 meters. And that's for a listener positioned on the narrow forward axis of a highly directional beam. Off-axis, substantially less. During the capture phase, when the whale is closest to prey, source levels are already reduced by 20 to 40 decibels below maximum. The effective acoustic exposure that prey experiences during a sperm whale's final approach is well below the source-level headline and, by Faze et al.'s calculation, below debilitation thresholds for squid and fish. The injury mechanism exists in principle. The demonstrated gap is between that principle and the specific claim that sperm whale clicks at natural interaction distances and with the behavioral output reductions documented during predation have ever injured or killed any organism through acoustic pressure alone. That gap hasn't been closed. Verdict: unsupporting the available evidence. This label does not mean the claim is physically impossible. It means there is no peer-reviewed documentation of sperm whale clicks causing injury or death in any organism at naturally occurring exposure levels. And the research specifically designed to test this, the Madsen 2006 debilitation study and the Faze 2016 tag study, found no supporting evidence. A claim that remains undemonstrated after targeted investigation occupies a different category from one that is merely untested. The four verdicts taken together: communication function, established; echolocation function, established; orca deterrence via clicks, specifically, plausible but unverified; injury and lethality, unsupporting the available evidence. What happens when a real and remarkable number, 236 dB are 1 micropascal, RMS, at 1 meter, back calculated, in underwater pressure units for a narrow directional beam, moves through social media without its units, without its measurement standard, and without its conversion context, is that it becomes a different number entirely. The original claim, which is already extraordinary, gets replaced by a simpler and more violent version of itself. Sperm whale clicks do multiple jobs simultaneously. Zimmer's 2005 beam pattern analysis shows that a single click produces a narrow, high-frequency forward sonar beam and a broader, less directional component in other directions. Two functional signals from one physical event, one for echolocation and one potentially useful for conspecific communication. The clan structure that Rendell and Whitehead mapped across the Pacific and Caribbean, built on learned dialects transmitted across generations, is built on the same anatomy that detects prey echoes from 245 meters. Flatten that into acoustic weapon, and all of it collapses into one dimension. The orca evidence leaves one specific question open. Across all the prey species that killer whale populations have learned to target, fin whales, grey whales, humpbacks, various dolphin species, which gives orcas the most documentably compelling reason to disengage from an attack? The physical and acoustic combination that adult sperm whales present across multiple observed encounters in multiple ocean regions is the strongest candidate in the available record. But strongest candidate in the available record is a different claim from proven deterrent, and that distance is precisely the gap between what the science shows and what the viral version says it shows.